Welcome to Biology Made Easy. Today we are discussing plant growth in flowering plants. Welcome. Learning objectives: identify differences between primary and secondary growth, outline the processes of primary growth and secondary growth, identify the tissues of secondary growth and their importance. Well, plant growth in flowering plants involves primary growth and secondary growth. So, what is primary growth? Primary growth forms the primary plant body, which is stem, root, leaf, increases the height of these and produces branches and lateral roots. So we have this plant here, a very young one. That is the primary plant body. It's the stem, leaves, root system. See some branch here, the same thing here, the stem, the leaves, root system. This is the primary plant body that shows primary growth. Secondary growth comes in to increase the width of the tissues that have been formed by primary growth. So it increases the width here, becomes bigger and tougher, strengthens the tissue. That is secondary growth. Well, is primary growth and secondary growth very separate phases in plants? Well, no. Primary growth produces the primary plant body, increases the height. While that is happening, the parts where primary growth has ceased thickens, goes through secondary growth to thicken, to widen, and to make it tough. So secondary growth will be going on in older parts of the plant where primary growth has ceased. At the same time, primary growth is taking place to produce new primary tissues. So the two go together in plants. Now, how do you recognize primary growth and secondary growth? Primary growth shows itself as soft green tissues. So it's herbaceous. Secondary growth, when it appears, shows itself as brown tissue and is tough and hard and gives strength. So in this plant, these parts have an undergoing secondary growth, while these parts are in primary growth. Well, primary growth and secondary growth takes place in plants. But note that primary growth occurs in all plants, monocots and dicots, right? Secondary growth occurs in only perennial dicots. So what are the tissues that produce primary growth? These are the meristems, and the meristems are at the apical meristems at the shoot tip, and these apical meristems at the shoot tip produce the shoots, primary tissues, stem, leaves, and some branches. So you have the apical meristem, you have the axillary buds also producing branches in primary growth, and then you have intercalary meristems. These are meristems at the node of monocots. They go to lengthen the internodes. Then secondary growth goes on in lateral meristems. The lateral meristems are the vascular cambium and the cork cambium. Now, the cork cambium develops very close beneath the epidermis, and then the vascular cambium is in here. These are the tissues that produce secondary growth. Well, notes should be taken in primary growth in monocots. As the whole plant lengthens, the internodes also lengthen. And in monocots, the internodes lengthen. This is one internode here. It will lengthen because there are intercalary meristems. There are meristems at the node here. That will cause the node to lengthen. This node will lengthen. There's this one at this node is intercalary meristems that will cause the node to lengthen. Let us now look at the primary growth process. As we said, the primary growth is brought about by apical meristems at the shoot tip and at the root tip. Now, the meristems at the shoot tip are found in the plumule. They are found right in the embryo, in the plumule here, and then the meristems at the root tip are found in the radicle. That is the source of the meristems. So the radicle will lengthen and produce the root system, and apical meristem will be left at the tip to continue lengthening the root, and also to continue producing new tissues of primary growth. Then the plumule will also produce the shoots, the stem with the leaves, and will leave a meristem at the shoot tip here to continue growing to produce more primary tissues and to lengthen the plant. So in primary tissue, you have a plant like this. This is a dicot. The same thing is happening in monocots. This maize, you have this part is the plumule. This part is the radicle. This radicle will grow and produce the roots and leave meristem at the tip here to continue growing to produce increase in length of the roots. And then here is the shoot. The shoot tip here will also have the apical meristem to produce more increase in length and more primary tissues of the shoots. So we want to look at the primary tissues that the apical meristems produce. And so if we have this dicot plant, the TS of it, section of it shows the primary tissues that goes on during primary growth. The primary tissues, the epidermis, the cortex, the pericycle, and vascular tissue. So if you come in here, you have the epidermis at the sides here. You have the cortex in here. This red band is the pericycle, and here is the vascular bundle here. And all this, and then the pith is here, and all this form the primary tissues. Here is it in my drawing. You have the epidermis, cortex, pericycle, vascular bundle. You have the xylem, you have the vascular cambium, you have the phloem, you have the pith. In between the vascular bundles, this area is parenchyma cell called the medullary ray. So medullary ray here, medullary ray. These are the primary tissues that is formed from the apical meristem that can be traced right in the embryo. If the plant reproduces vegetatively, then it will start from the buds. But how these primary tissues are formed, the detail of it is found in my lesson on plant tissues and how they are formed. So please find out, and you get a detail of what happened to form these primary tissues in primary growth. Well, this is also primary growth that occurs in monocots. And then you have here, if this is cut, a section of this is cut, you have the epidermis is here, you have sclerenchyma, you have these are the vascular bundles. This is parenchyma. Here is the dicots. I just put this here to show the primary tissues we will be dealing with in secondary growth, because secondary growth does not take place in the monocots. Secondary growth, as we have said, is also called secondary thickening, and it goes on only in the perennials of dicots and also in conifers. Now, it occurs in areas of the plant where primary growth has ceased and is brought by lateral meristems, as we have said, vascular cambium and cork cambium. Cork cambium is also known as phylogen. Then it produces, secondary growth produces the secondary xylem, wood, secondary phloem, bast, and then periderm. The periderm is made up of cork cambium, secondary cortex. Well, we should not forget that in here, there's also secondary medullary rays also in here. Right. At the onset of secondary growth, the vascular cambial ring is formed and cork cambium phylogen is also formed. Now, look at this primary growth. We have this vascular cambium. We don't have a ring of cambium, but at the onset of primary growth, a cambial ring is formed here. So we have a cambial ring formed here. And then also, look here, we don't have cork cambium here, but then we have a cork cambium appearing here. This is at the onset of secondary growth. How these forms, we will see soon. Here we have the vascular cambial ring, how it was formed. Look, you have parenchyma cells here, parenchyma cells in between here. They become meristematic and divide. And then the vascular cambium here too, some of the cells, also the cells divide, and some divide radially. So the two divisions of the parenchyma cells here and the vascular cambium here forms the cambial ring here. Here is it, all right? Vascular cambium forms, and also the cork cambium also forms. The cork cambium forms as parenchyma cells beneath the epidermis become meristematic, divide radially, and forms the cork cambium, also known as phellogen. So that is how these cambial rings form. Now, let's see what happens after the cambial rings have been formed. Now, here, the vascular cambial ring, as well as the cork cambium, they all work to divide and build tissues at the same time. So here, the vascular cambial ring, it has two types of cells, fusiform initials and ray initials. This vascular cambium here, ring, it has two types of cells. And the vascular cambium divides tangentially to produce secondary xylem in here and secondary phloem out there. And then also it has cells that will produce radially to form medullary rays. Okay, so that is what the vascular cambial ring does. It forms here, it forms secondary xylem in, that is wood, and then medullary rays, growth of parenchyma in the secondary xylem, which is wood. Now, then it also forms secondary phloem, that is the bust outside. And so the primary phloem is left at the back here, and this primary xylem in here, and the pith. Whilst the vascular cambial ring is doing its division to produce the secondary xylem and the secondary phloem and the secondary medullary rays, the cork cambium will also be dividing at the same time to produce a layer of cork, which is phloem, outside, and then a layer of secondary cortex in there. So the two cambial rings divide at the same time to produce these tissues. So you have the secondary cortex is called phloem, and the layer of cork is phloem in here. That's what the cork cambium does. Now let's look at what happens further developments as we study this very carefully. The cork cambium, phellogen, produce the layer of cork, which is phloem, and secondary cortex, and then the vascular cambial ring produces the secondary xylem, secondary phloem, and secondary medullary rays. Now, look at it carefully. The primary phloem is here, and the primary phloem is weak. And so normally the primary phloem is finally crushed, and only the secondary phloem that is being produced is kept. And then you have the primary xylem also stuck in here, and then the pith. Now look at the difference between the primary medullary rays. We've labeled some. This is a primary medullary ray. This is a primary medullary ray. The primary medullary rays, they were formed at primary growth, and they were the medullary rays that were connecting the pith to the cortex. That is the primary cortex. So this is a primary medullary ray. This is still primary medullary ray. You see, it's connecting the pith here, and it goes back here, so it connects to the primary cortex. Now, this is secondary medullary ray. The secondary medullary rays, they are formed only in the tissues that were formed in secondary growth. So this is a secondary medullary ray. This is also another secondary medullary ray. So take note of that. Good. As secondary growth continues to go on, this is what happens. The epidermis, which cannot grow and give, peels off. starts peeling off. So the epidermis is peeling off. The cork layer is becoming thicker. This is still cork cambium and secondary cortex, secondary phloem. And you have, look at it, primary phloem. This is cork cambium. You have secondary cortex. You have primary cortex. This is primary phloem. This is secondary phloem. This is vascular cambial ring. And this is secondary xylem wood. And the primary xylem is here and the pith. Now, look at the medullary rays stuck in there. And as you can see, more xylem is formed more than phloem. Looking here, we have the cork here. Just want to look at the picture here and look at it in the tissue here. So you have layer of cork here, right? Then we have the cork cambium here. Here is the secondary cortex. Here is the primary cortex because you can see some pericycle in here. Pericycle is red, all right? Stains red because of thickened tissues. Then you can have some phloem here. And here is the vascular cambium. And all here is wood, secondary xylem. You have the pith here. Now, let's find where the medullary rays are. Look at this. This will be a neat secondary medullary ray. It's only in the secondary growth. This also a secondary medullary. This one is a secondary medullary ray. But look at this. This is a primary medullary ray. It's connecting way down here up to the cortex to the pith. So that is the primary. This is also another primary medullary ray. Well, let's see what goes on as more secondary growth goes on. The epidermis is completely peeled off in here. You have a Lenten Sole here. Those are holes through which gases move in and out of the plant. We have our cork, which is phloem. We have the cork cambium here and some secondary cortex, primary cortex, secondary phloem here. Primary phloem would have been crushed up here. And here you have wood, secondary xylem. In here, you have wood that is lighter in color. That is the sapwood. It's still living. It will still have a lot of medullary rays, that's radial conduction through this sapwood, which is living. And you have wood, heartwood here, which is dark and normally very dead because it's filled with tannins. So it's strong, very strong and thick to support the plant. Well, then you have these. This is an annual ring. This is an annual ring. This is an annual ring. The annual ring shows the amount of wood that is formed in 1 year. And you normally have a thinner layer and a darker layer. The thinner layer showing areas that the wood was formed in rainy season quickly. And then you may have a darker area of xylem that was formed slowly in dry season. So we look at the stuff here, a section here. You see this is cork and this is living tissue here. You may have, so you have the cork here, and then you have all here is living tissue. You have cork here, you have cork here, you have cork cambium here. Cork cambium is here. You see the cells here. Then you have all here is living tissue, can be secondary cortex and primary cortex and secondary phloem, all here, this living tissue. And then you can have in here some vascular cambium here. Look at it. All here is living, from here, cork cambium, living tissue, and then you have the vascular cambium. So at the back here, all here is. So here is our annual rings. This one, annual ring, annual ring, annual ring. All right. And we have secondary medullary ray here. We don't see any primary medullary ray yet, but that is it. The pith has been crushed. It was here. Good. So let's look at what is happening in here. We have the cork here, layer of cork here. This is our living tissue here. We have our cork here. We have our periderm here. And then we have the vascular cambium in here and wood. So everything behind the vascular cambium form the back. As you can see, the back is here. Look at this. Tissues that form the back. That is it. And we come here. This is the back. The vascular cambium is somewhere in there. This is an old cat wood. All this part is living, would have been living whitish. And this is the cork. All this shows the annual rings in there. Good. So we are going to look at what a Lenten sole looks like. You have, this is the hole. These are loosely packed parenchyma cells so that air can move from outside the plant in here and get into the living tissues, the cork cambium, phellogen, and supply the plant with air, oxygen, and carbon dioxide in. The same goes out when it's necessary. That is the Lenten sole. So you can see the areas that form it, loosely packed parenchyma cells. You have the epidermis, which is peeling of cork cells, cork cambium, secondary cortex, primary cortex. And these are the names of these tissues, right? So let us sum up as we look at the structures or the tissues, as we look at the tissues that form secondary growth. You have heartwood. Soha is dead and strengthens the plant. Sapwood is whiter. It has living tissue. It strengthens the plant, transports water and mineral salts. Bast is secondary phloem that transports manufactured food. Medullary rays, you see where you saw them. They do radial conduction, mineral salts and water. Gaseous exchange because they are parenchyma cells. They also store food, especially during dormancy. You have the cork phloem, which protects the tissue from losing too much water, from infection, from mechanical damage, and it replaces the epidermis as the epidermis peels off due to increasing gait. Then you have the Lenten soles, as you have said, pores, all right, that allow gases in and out. And then we also mentioned the annual rings, wood formed a year, made up of soft, wider xylem vessels produced in rainy season, and harder wood or small lumen made in dry season. And it shows the climate of the area, and it can also show the age of the tree. So this is what we have for today. Thank you and bye-bye.